All right, get the lights right. Okay. So before I get to real science, we have a little bit of sort of course-type stuff to touch on. Let's see now, here we go. The miscellaneous things we talked about last time, I just want to make sure that they're 100% clear to everyone and that kind of thing. I'll just use a scroll bar. There we go. Just reminding folks that there's a study group on Sunday night. That'll be 6 to 8 p.m. Review session Monday night. A point that we did not announce on Wednesday is that Susan has needed to reschedule her office hours. Doesn't like to do it, but she's presenting at Research Day, and so certain things are compelling. So I rearranged Wednesday, 2 to 3, same place. Reminder that the class is going to move. If you turn up here, it'll be good science. It won't be our biochemistry. It'll be different biochemistry, and I'll show in a second a map with that. And just one last reminder that a week from today will be the next hour exam. And then you should have received this file. Did anybody not receive this file? If not, you might want to check on your campus address because it was sent to whatever one the university thinks we have. So the General Hospital is the great big complex, and the part we're interested in is the northern part near Boyd Tower. And more specifically, okay. I'm so used to it doing things to me, I expected it to fail again. It's right about up in that quadrant, I'm pretty sure on the 3rd floor, and one of our intrepid explorers is going to try and find the right elevator for you. Probably elevator A. I don't want to swear to it in advance, but probably elevator A. Not a bad room, just not ours. Any other questions about the relocation Wednesday before we move on to real science? Okay. Do people have any questions about here we go, let's get this closed off. Do people have any questions about lipid metabolism before we move on to nitrogen metabolism? Just go away. All right, then let's move on to nitrogen. So this is the last of the great 3 categories. Carbohydrate metabolism, you're dealing with things that are half oxidized, half reduced. Lipid metabolism, less variety in a sense because there's fewer atoms involved, but more reduced. But in some senses, a larger spectrum of sizes and shapes. And now we're going to get into nitrogen metabolism, and nitrogen metabolism probably has the greatest variety of structures, of reactions, of all of them. And we're not going to make any attempt to cover the whole of it in this class. It's a semester worth in its own. What I will try to do is see to it that you feel comfortable with the most important concepts of it, that you have a good sort of landscape view of the key elements of nitrogen metabolism. Now, nitrogen is present as an atom in 2 big classes of molecules, the amino acids and the nucleotides. So it's playing a really, really crucial role. Nitrogen is needed for information, and nitrogen is needed for catalysis. And those are 2 of the reasons why nitrogen metabolism is so important, even though so weird. Interesting, weird. We're going to see that nitrogen in the air, which is N2 with a triple bond, has to get fixed into some kind of organic form, and that's energetically expensive. It gets used to be put into amino acids, and it's going to eventually get put from there into nucleotides. Sometimes you actually have a superabundance of it, and in those cases you have to get rid of the extra nitrogen, and we'll see how we do that. And if there is an excess of nitrogen, that leaves a variety of carbon backbones, and then we start talking back over there towards carbohydrate metabolism and lipid metabolism. And we'll pick up our pointer. I should make it possible to set a default on this. Okay. Nitrogen fixation is a really challenging biochemical event. Nitrogen itself has a triple bond like that, and it's a very, very stable inert bond. And so trying to do anything with it, it's very difficult to do energetically. The availability of nitrogen is one of the principal limits on how much life can grow somewhere. Phosphorus to some extent, in the ocean iron, but certainly nitrogen is one of the big ones, and that's why people have been spreading manure on farm fields for millennia. There are only a few organisms that can do this remarkable feat of taking molecular nitrogen out of the air and making it organic, or at least accessible. Blue-green algae are a real big one, cyanobacteria, and they have been playing a critical role in agriculture ever since rice started to be cultivated, because the nitrogen in rice paddies comes from blue-green algae, and also some bacteria. But it's not a universal trait by any means. And it's accomplished by this enzyme nitrogenase. Really, really unusual. I believe it has only evolved once, as far as we can tell. The energy of it is, the energetics of it and the chemistry of it are so strange. It has a mixture of iron sulfur and iron molybdenum cofactors. So here's a picture of that structure with the iron sulfurs, and there's your molybdenum. And ferredoxin, of course. Ferredoxin was our handy-dandy reducing agent from photosynthesis. The marvel, or the miracle of nitrogenase is it can do at room temperature what industry needs to have 400 degrees Celsius and 300 atmospheres. So this is an absolutely stunning enzyme. And what it's going to have to do is along these lines. We're going to start with molecular nitrogen, and it's going to consume protons, electrons, 16 ATP, and 16 waters to produce 2 molecules of ammonia and the byproducts you might expect out of that. A little bit of hydrogen gas, interestingly, too. This is the optimistic estimate. In some cases, it's as bad as 20 to 30 ATP for every nitrogen fixed. So this is a difficult reaction. It takes so much energy that tells us it's a difficult reaction, and it's one that no one bothers to do unless they have to. It's also one that's extremely sensitive to oxygen. This complex, as you can imagine, is just ripped apart by oxygen. And so nitrogen fixation happens only in anaerobic circumstances, which further limits what's going to go on. And it turns out that there have to be tricky ways of getting the nitrogenase anaerobic in order to make it happen. It's a leftover from the time when Earth was anaerobic. It goes back that far, and no one ever found a way to do it in the presence of oxygen. So very unusual chemistry. Here you can see some nodules. So this is a clover plant, obviously macroscopic, but these little bumps on it are some of these bacteria that are capable of doing the nitrogen fixation reaction. And of course soybeans have that. That's why soybeans are such a marvelous thing. Now there are also other ways of getting nitrogen if some of the other forms of nitrogen are available. So for instance, an enzyme nitrate reductase can take nitrate ion, NO3, and add electrons to it, and take that down to NO2, which is nitrite. Nitrite reductase can take NO2 and add a lot of electrons to it and get ammonia. So these are two reactions that are consuming electrons, but it's not nearly as expensive as trying to fix it right out of the air. And the end of it all is ammonia, which we're going to see is a very usable form of nitrogen for biological purposes. And these reactions are much more common. So this is consuming electrons, and as you can imagine, you have to invest energy to get that to happen. Nitrification is going in the other direction, from ammonia to nitrite to nitrate, and there are bacteria that make their living that way by being a nuisance and making the ammonia less useful for us, and denitrification, which takes it down in that direction. So there's a cycle, or not really exactly a cycle because some parts go in other directions, but there's interrelationships amongst all of the different forms of nitrogen, and these things are present in the environment, in the soil, sort of a constant commerce going on in nitrogen of one form or another. So what are we going to do with the ammonia? In order to take ammonia and really make it organic, ammonia itself is simply a reduced form of nitrogen. But to make ammonia in an organic form, we have to then combine it somehow with a carbon backbone. And this is done through glutamine synthetase. This is an enzyme that every organism has. One should hardly ever say ever. Every organism I know about has. There are probably some parasitic ones that still lack. But it's very, very, very widely spread, and it's a place where nitrogen can enter actual metabolism in a biological form. It's also a way of environment. That is, if there's ammonia left over from other reactions, it's a way to get it out of solution and into an organic form that's not damaging. Ammonia is a base. I mean, you all use, I assume, you all use Windex on your windows at one point or another. That's ammonia. And it's very tough on the dirt on the windows, it's very tough on cells. So free ammonia is not a good thing. And glutamine synthetase is one way of getting that free ammonia out of the way and into something more manageable. And this is what that overall reaction is going to look like. So here's the ammonia here that we want to add. And in order to get it there, we have to activate the glutamic acid. So if we look at this carefully, this is a glutamic acid. Here's amino acid end here. This is the glutamic acid end, 2 carbons, glutamic acid. We invest an ATP and activate it. So now there's a phosphate group at that end. And as you can imagine, this group is more reactive than it was before. And so this group is capable of being a site of attack by ammonia. The ammonia comes in, inorganic phosphate comes out, and now we have glutamine instead of glutamate. And so this reaction has cost us an ATP to get to that point. So clearing one molecule of ammonia out of the environment costs us 1 high energy bond. Glutamine, as an amino acid, is one of the most abundant amino acids. In many cases, it's many times more abundant than any other amino acid. And that's because it functions as a nitrogen carrier in the body. Some tissue culture cells only grow if there's a vast excess of glutamine in the solution and so on. For that reason, the glutamine synthetase is regulated, so it's not scarfing up everything all the time. This does give us sort of a regress thing. For glutamine synthetase to work, we had to have glutamate. And glutamate already has a fixed nitrogen on it. So where did it come from? Well, you may ask. We can make another molecule of glutamate if we have, to start with, a carbon backbone like alpha-ketoglutarate. We have a glutamine molecule to act as the nitrogen donor, and a source of reducing power, in this case NADPH. And what's going to happen is that the carbon up at this end is going to become reduced a bit, reduced enough to become an amino group, and the nitrogen that was down on the glutamine is now up where there was a carbonyl oxygen before. So when you start off with one molecule of alpha-ketoglutarate and one molecule of glutamine, which has 2 nitrogens, you end up with 2 molecules of glutamate, both with their amine groups in the alpha carbon like any other amino acid. So this is a reducing oxidation reduction reaction, and it provides more carbon backbones to scarf up more nitrogen from the atmosphere if we need to do that, or more molecules of glutamate for protein synthesis. So, any questions about that before we go on? Okay. Now that we've gotten this nitrogen up into the amino acid end of the molecule, we can move that nitrogen around quite easily. In fact, we can move it around for free. We've got it up in a form chemically that is going to be equivalent to its form in many other places, and so it can just simply be switched around through enzymes that can accomplish that. So if we take, for instance, an amino acid, an alpha amino acid, and an alpha keto acid, so this is the alpha carbon, this keto acid, transaminases are enzymes that can simply move the amino group from that one to that one and recreate a carbonyl oxygen over there. So transamination reactions are completely free, they're freely reversible, and they allow you to move nitrogen around from one carbon backbone to another. Glutamate is often one of the donors, So it provides a pool of nitrogen to make other amino acids. And it's not uncommon to see citric acid cycle intermediates and some of the glycolytic intermediates as the receivers that are turned into amino acids. This particular transamination reaction gives you back alpha-ketoglutarate, which is, of course, a citric acid cycle intermediate, and alanine, one of the simple amino acids. It's a very lovely trick, and it's accomplished with the help of pyridoxal phosphate. Pyridoxal phosphate looks rather like this, and you may kind of remember the sound of it because occasionally vitamin pills spell it out. It's vitamin B6, pyridoxine. So here's pyridoxine, and here's pyridoxal phosphate. You can see places that will sell you the activated form with the phosphate on it. Don't waste your money. The moment that this stuff hits the stomach acid, the phosphate's gone. You might as well have bought pyridoxine. And besides, your body's perfectly capable of putting the phosphate on. Because this is a common cytoplasmic enzyme, it is released if cells die. And so transamination enzymes are markers for tissue damage. And if you've ever, God forbid, are at risk for a heart attack or cirrhosis or something, they will look for the presence of these enzymes in your blood to see if there are cells that have died and let them go. And part of why the pyridoxal is so common is it can form a Schiff base as a way of holding on to the intermediates of the reaction along the way. So let's take sort of the above view of some of this metabolism. Let's look specifically at the map now in terms of amino acid synthesis. And so amino acids and nucleotides are themselves the monomers that are going to be used for other things. We'll be talking later on about how we make nucleic acids. We'll be talking later on about how we make proteins. Those are both part of macromolecular metabolism or macromolecular biology, last part of the course. Let's just focus now on sort of the simpler level of these organizations. We're going to need to make amino acids, we have to have carbon backbones. And the principal places that we're going to get those carbon backbones from are the citric acid cycle, glycolysis, and the pentose phosphate pathway. These are three different places where carbon backbones get rearranged in various ways, anywhere from 3 to 6 carbons long. And so they provide us with a lot of variety for what we can make amino acids out of. The nitrogen that's going to be needed for amino acid synthesis, as I mentioned already, is often, not always, but often, from glutamate or glutamine. And we can make 10 of the 20 amino acids of the standard 20 that go into proteins. There are a few other weird ones. We call the 20 that we can't make essential amino acids because they have to be in the diet or we're sick. The other 10 amino acids are every bit as necessary for life, but they're not essential in the diet. All of them are needed to make proteins. Essential amino acids must be in the diet. Now, if we look at the particular amino acids that are listed, the non-essential amino acids are things like alanine, aspartate, asparagine, cysteine, glutamine, glutamate, glycine, proline, serine, tyrosine. They are often the smaller ones. There's often one of a group. So for instance, we've got cysteine here, we'll see methionine over there. We've got tyrosine over here, we'll see phenylalanine over there. And so there's a sort of a suggestion that we might have kind of some building up from one or the other. And over here we have the larger and weirder ones, okay? Isoleucine and leucine, arginine. Arginine is quasi-essential. You and I probably don't have to have it in our diets, but people who are still actively growing in significant amounts, vertically, not horizontally. Horizontally, you just need carbon backbones. Growing vertically, you need arginine because the liver can't quite keep up. And we're going to see why some of those things are tryptophan, huge, complicated, weird ones. So these are all things that are hard to make, and we'll see some of why. Okay. Now for the non-essential amino acids, the reactions that make them are surprisingly simple, and there are certain patterns that recur fairly frequently, and so they're worth taking a little look at. Let's say we start with an aspartate fairly simple amino acid, one carb. It's basically an alanine with a carboxylic acid. We can make aspartate into asparagine using a nitrogen from glutamine and some ATP. And so the nitrogen that was on the glutamine is going to be incorporated down in that peripheral carbon, so we now have an amide group instead of carboxylic acid. So the glutamine has become glutamate, and it's cost us 2 high-energy bonds. We can make some other ones through transamination. If you think about the structure of these molecules, you'll see that pyruvate is transaminated to alanine. Oxaloacetate is transaminated to aspartate. Alpha-ketoglutarate is transaminated to glutamate. You just have to look at the structure of those molecules to see that that would be true. I always feel sorry for the people watching the video when I cough. So we've seen how aspartate can become asparagine through asparagine synthetase. Same kind of logic for glutamine synthetase if we want to start with glutamate and make a glutamine. So we've seen a couple of ways of making glutamine here. And then, so if we add all of this up, you can see that just 3 citric acid cycle intermediates can give us 5 of the 10 amino acids by very simple reactions. So we're a quarter of the way home. Some other places there you see sort of amount of logic to what's going on. Glutamate has a 5-carbon backbone. We've mentioned it going to glutamine. The carbon in the carbon backbone of glutamate through several steps, that's why it's a dotted arrow, becomes the 5 carbons of an arginine backbone. And you can see that this structure might be complicated, and that's why we don't make a lot of it. And the 5 carbons, this backbone through several steps, become the funny kinked closed circle of the proline backbone. But these pathways are somewhat more complex. You can imagine just by looking at these things that it's not 1 or 2 steps to get from glutamate to those things. But now we have 8 of the 10. Serine is kind of a funny one. Just to refresh your mind on serine, it's just alanine with an alcohol. It doesn't look like it ought to be particularly fancy. But we don't start from there. We start from a phosphoglycerate. We're going to reduce this so that this alcohol is going to become a carbonyl carbon here, a keto group. So this was an alcohol. There's a double bond oxygen, so this is clearly more oxidized. Now we've got the NADH off of it. This is going to become the recipient for nitrogen. You remember this is an alpha keto acid, and so it can be transaminated. When you transaminate 3-phosphohydroxypyruvate, you get 3-phosphoserine. So the glutamate is going to donate the nitrogen by transamination, and now you have 3-phosphoserine, and all you have to do is throw away that phosphate group, and now you have a serine. So in a sense, we start from 3-phosphoglycerate as a pre-activated intermediate, if you like. But of course, 3-phosphoglycerate is a glycolytic intermediate, or if you like, a pentose phosphate intermediate. Glycine, you think should be very simple. It's actually a little bit odd. Glycine starts from something bigger than itself. Maybe that's not so odd. Glycine is a 2-carbon fragment, which is kind of odd in its own right, with a lot of stuff hanging off. It starts from serine. We just saw serine structure, and it has 1 carbon too many. So that carbon is going to be removed through tetrahydrofolate. And tetrahydrofolate is going to reappear many times. Tetrahydrofolate is a very good cofactor for moving carbons around. And it can move carbons around in a number of different oxidation states. And you may hear in it folate, it's folic acid. It's why you need your dark green vegetables, and it turns out to be a good way to prevent birth defects. But this reaction is actually reversible. So if you have extra glycine, you can make serine, and if you have extra serine, you can make glycine. But now you have your glycine, and the carbon is up there now. I guess I was kind of giving away the punchline to the story before I got there. We see tetrahydrofolate not just in amino acid metabolism, but also in nucleotide synthesis. For both amino acid synthesis and nucleotide synthesis, we have to change carbon backbones a fair bit. and tetrahydrofolate is a good carrier for carbons for that. It got its name because it is in leaves, but it's also in some vegetables or in germs, like wheat germ, stuff like that. If it's deficient in pregnancy, a condition called spina bifida can occur, where the backbone of a baby is not completely closed over. And why that should be true, we don't understand. But it's a very strong association. It's why baked goods are supplemented with folic acid here and in most parts of the world now, with a huge decrease, 80 to 90% effective at reducing the incidence of spina bifida. This is just showing you one of the several forms of tetrahydrofolate, and here it is with the carbon atom in the methylene condition. Here's the whole of tetrahydrofolate, and the sort of the business end, if you like, is in here, where it can undergo various states of oxidation and carrying carbons. There's a paraminobenzoate here. You might have heard of that as PABA, and a tail of glutamates there that act as sort of the tail to hold on to it. We can do most of it, but we have to have, actually no, we need to have this in pretty much this shape to make it work. Microorganisms can do most of it except for the PABA, and so if you have antagonists to this, you can inhibit bacteria without inhibiting us. What about cysteine? Cysteine is a sulfur-containing amino acid, and we need to think of how we're going to do the cysteine. If you start with a serine, that's kind of promising. If you were to have a molecule of homocysteine, which is like cysteine but with an extra carbon in there, you can then come over and use this sulfur to attack that carbon, and you end up with this odd kind of, cross-bridged thing that's got two amino acids connected through a sulfur called cystathionine. And if you can afford another nitrogen from somewhere, you can, if you remove one of the nitrogens, you can then end up with a proper cysteine here and alpha-ketobutyrate. It's alpha-ketobutyrate, not glutarate, so it's the 4-carbon, not the 5-carbon. We've talked then about the non-essential amino acids, the ones we can make. Those pathways I've talked about so far are in our bodies. The essential amino acids, all of them have either complicated pathways or expensive pathways. And they are present in some animals, all plants, but we're missing them. And one way to remember what we can and cannot do, we in particular can't make branched chain amino acids. So valine is branched, leucine is branched, isoleucine is branched. Threonine is a branched chain if you think about the fact that the alcohol sticks off the side. Okay, you can think of the methyl group as the branch. We can't make aromatic amino acids from scratch, phenylalanine or tryptophan. We can't fix sulfur into methionine. So those are all going to have to be present in the environment. Why is this thinking? All right. How does the sulfur get into organic form? We talk about fixing nitrogen. Well, sulfur's out there in a variety of forms. How does the sulfur get into organic forms? One of the places is through sulfide. Bacteria can do that. Plants can use sulfate. But we can't do either of that. Once you have a serine molecule, you can then take advantage of an acetyl-CoA like this to make an O-acetyl-serine. So this is your serine starting point, which you've acetylated here. So this oxygen there is in kind of an unusual configuration. And the reduced sulfur here is now able to attack at that spot, which gives you the cysteine, and it releases the acetic acid. Methionine is then going to also take advantage of homocysteine in a slightly different way. Here you have homocysteine, And it's going to need to have another carbon on it to become methionine. That's what its name means, methylated thionine. It's got a methyl group on its sulfur. So it's going to need a methyl group. So methyl tetrahydrofolate will donate a methyl group to add to there and give you methionine. This is one of the places where we see the effects of folate in the diet, that if there's insufficient folate in the blood, we see high levels of homocysteine. So in other words, to get from homocysteine to methionine, you need tetrahydrofolate. And the principal place we see metabolically the consequences of the low tetrahydrofolate is that this accumulates. And there are people in which there's a chronic deficiency in this who have extremely high levels of it. But even in people who don't have that genetic condition, as this accumulates, it predicts heart disease. Yet another reason to eat green vegetables. So where do we get, or I should say, for the essential amino acids, where do the carbon backbones come from? There are a lot of steps involved, and I'm not going to try to walk you through all of them, but to give you a sense of how connected these various pathways are. Aspartate, the 4-carbon amino acid, can donate carbons to the backbone of methionine and threonine and lysine. In animals that can make these molecules. Pyruvate, 3-carbon backbone, can contribute to isoleucine, valine, and leucine. Aliphatic amino acids are going to pick up their nitrogen at some point, and they do that from glutamate by a transamination reaction. The aromatic amino acids are kind of interesting because they have implications both medically and also for agriculture. To make things like phenylalanine and tryptophan, we have to make a molecule called charismate. You can see how it's potentially going to become an aromatic ring there. To get there, we start from phosphoenolpyruvate, PEP. This is our end product of glycolysis or an intermediate of the pentose phosphate pathway. Here's erythrose 4-phosphate, another pentose phosphate intermediate. And these two together, a 3 and a 4, can give us this 7-carbon molecule. And through very clever enzymes, phosphoenolpyruvate can be added then to that to give you the charismate. So this linear structure closes around into a ring with a bunch of spinach hanging off of it. And the reason that this is significant in terms of agriculture is that the target for glyphosate, which is Roundup, is this enzyme. Since we don't have this enzyme, it's not at all toxic, or essentially non-toxic to humans. In fact, it's not particularly toxic to most animals, because most animals don't make their own aromatic amino acids. And so it's a nice example of how biochemistry allowed for some more specificity, some more subtlety in the industrial chemicals we have. Tryptophan. There are 13 steps to make tryptophan. So it's not surprising that tryptophan is an essential amino acid for a wide variety of organisms. There are even bacteria that don't bother to make their own tryptophan. Also an amino acid that cells try not to use if they don't have to. So it's fairly rare in proteins. It's usually in strategic places. The last 2 steps are sort of cute, though, and I'll just sort of touch on them. You have this intermediate. It's an indole ring with a 3-glycerol phosphate. So it's got this glycerol phosphate hanging off of it, the 3-carbon. You can split off a molecule of glyceraldehyde 3-phosphate, and then that gives you indole proper. And then adding a serine to that gives you tryptophan. You can see that that's quite a cute trick. Serine and the nitrogen, the serine provides the amine group up there. Now, what's funny about this particular reaction that goes from indole 3-phosphate to tryptophan is that it occurs on a protein that has 2 enzymatic activities. These 2 enzymatic activities are completely different, right? Splitting off glycerol 3-phosphate and adding a serine really are not similar at all. One's happening at this carbon, and one's happening, I guess it's happening at the same carbon again, but very different kinds of reactions. And the enzyme, tryptophan synthetase, is a bifunctional enzyme, and the indole stays on the enzyme but tunnels its way from one end to the other. So it starts down at this end, and the indole then sort of squirms its way up to here, and the second reaction happens up at the other end. So it's a nice example of how enzymes are really capable of extraordinary structural adaptation to their demands. This makes sure that the indole doesn't go anywhere and doesn't get lost in any way. What about histidine? Histidine's pretty weird. Here we go. There's the structure of histidine. So it's alanine down to there, and then it's this odd 5-membered ring with a couple of nitrogens in it. This is the only amino acid that gets some nitrogen from a nucleotide. And it makes a little bit of a difference to talk about it because it turns out it's an important intermediate. If we look at ribose triphosphate, so here is a ribose triphosphate. That can become 5-phosphoribosyl pyrophosphate after a number of reactions. So, And so this molecule, PRPP, as it gets called for short, okay, it's P-ribose PP, so it's a ribose with a lot of phosphate hanging off it. PRPP is important because it is not just used for histidine synthesis, it's also used in nucleotide synthesis. So it's one of the few places where it's overlapping between the two domains. And some people think that it's sort of an echo of the RNA world's past. At any rate, so PRPP is going to become the platform on which we finish building a molecule of histidine. And both glutamine and glutamate are involved in contributing the nitrogens that are going to go into this. You've got to get a nitrogen up there, and you've got to get nitrogen into those rings as well. We've thought about amino acids mostly so far as building blocks for proteins and their structural properties. And we've talked a little bit about them as carbon backbones for gluconeogenesis. But there's also a role they play that's completely different from those. Many amino acids are neurotransmitters. We talked about exocytosis of neurotransmitters back when we were talking about membranes and action potentials. Some amino acids are neurotransmitters just plain and simple. So glycine, glutamate, and aspartate, each of them act as neurotransmitters. There are receptors in neurons that are gated by the binding of the amino acid. So they will turn on or turn off neurons directly. They are also precursors for some other neurotransmitters. So if you take glutamate and you take off a CO2 from it, you get GABA, gamma amino butyrate. So methane, ethane, propane, butane. So this is a butyrate, and it's gamma amino butyrate because it's on the gamma carbon. We call it GABA. GABA is one of the major inhibitory neurotransmitters. That is, it's just as important to make sure that neurons don't fire as it is that they do fire. And GABA is one of the major players to keep things from firing too much. Some of the other ways in which amino acids contribute to making the whole spectrum of transmitters we need. Tyrosine, for instance, can be decarboxylated again to give you dopamine. You also have to add a hydroxyl group there. Dopamine is especially important because of its role in Parkinson's. Parkinson's is a degenerative disease in which the lower levels of dopamine progressively lead to less and less ability to move, especially the muscular framework of the body. It doesn't so much inhibit thought as it does the ability to move and express it. And a beautiful book and movie called Awakenings discusses the response of the first patients to dopamine when it was first discovered. Tyrosine is also going to give rise to derivatives norepinephrine and epinephrine. I think you can see the kind of relationships amongst these. So catechol has had an extra alcohol added, and then it's the building frame in which you add spinach on the top. Epinephrine and norepinephrine, of course, are involved in response to stress. They may play a role in depression. Epinephrine and norepinephrine reuptake inhibitors are among the major classes of antidepressant drugs. Okay. Great. Let me just take a quick peek to see where we are. This is 23, 2. Before I get too far into this, I want to make sure I say something before I'll have to leave. I hope you picked up the single sheet that was out there. The single sheet is a study aid that Angie put together, so you can thank her the next time you see her. She had to go to an event today. And it's a nice comparison between mitochondria and chloroplasts. So if you were fuzzy about things, you have a nice place to turn to sort of refresh yourself on those. Continuing with amino acids on the neurotransmitters. Tryptophan, here we have it, which has 2 rings, heterocycle there, big bulky thing. It yields another important neurotransmitter, serotonin. You can see it's another decarboxylation and an alcohol, sort of a recurring strategy there. That's serotonin, and serotonin is another amino acid that's neurotransmitter that appears to be involved in depression. Seems to play a role in sort of one of the psychiatrists who's written engaging about it calls it the policeman of the brain to kind of keep things in the right margins. Prozac is a reuptake inhibitor that keeps the levels of serotonin higher in neurons. I feel compelled by honesty to point out that although I'm describing these neurotransmitters, what are called biogenic amines, as playing a role in depression, it's quite possible that the antidepressant effect of these things is secondary because they have their effect at the neuron endings within hours, but most of them don't have their antidepressant effect for days or weeks. We don't really understand how antidepressants work, but there is clearly some kind of relationship to these neurotransmitters, and these are clearly neurotransmitters that play very important roles. And then serotonin itself, through another couple of rearrangements, gives us melatonin. And melatonin is quite a nifty molecule because it plays a role in circadian rhythms and how we can get re-entrained each time to the fact that the day is turning into night and so on. So, another very odd kind of a donor for something that's playing a role is arginine. So one of our biggest amino acids can give us nitric oxide, which is the smallest neurotransmitter. You can't get smaller than 2 atoms, right? How it goes is something like this. You have arginine. It's got that spinach at the end of the guanidino group. If we reduce it and use oxygen in the process, we can make a hydroxyarginine. So here's a hydroxy group hanging off it now. And if we reduce it further, we end up with citrulline. We're going to see citrulline again later. Remember citrulline. But having made this, one of the other products is this nitric oxide radical. That little NO over there is the actual neurotransmitter. And it is a very important one. We're finding more and more systems in the body that are regulated through nitric oxide. Muscles respond to it, heart responds to it, the immune system responds to it. Nitroglycerin here has been known to calm down angina, the pain in the chest from overconstriction in the heart since the 19th century. It turns out that it's giving up nitric oxides. And it's a nitric oxide mediated system that Viagra works on, although not on this particular part of it. So. Any more questions about amino acid metabolism, amino acids for neurotransmitters, anything like that before we go on to talk about nucleotides? Oops. Let me just see if we can risk calling that a day or not. I think we can call that a day if you'd like to head off for the weekend. Have a lovely weekend, despite the rain.